Alright guys, so here we are at Concept 6.2. Concept 6.2 says eukaryotic cells have internal membranes that compartmentalize their functions. I think if you'll think back to our very first assignment where we were listening to that TED Talk and Adam Savage called us just bags of meat or bags of water or something similar to that, it's actually fairly accurate. We're going to be looking at eukaryotic cells, which are the types of cells that make up our body and most other living plants and animals. And not just bags of water, but you'll find that our bodies are really bags of water within bags of water within bags of water. So we're going to drill all the way down to the cell parts, and we're going to look at these organelles, and you're going to find that they are basically bags bound by membranes within a cell that is bound by a membrane within our own bodies, which are bound by our skin, which is another type of membrane. So you're talking basically about bags of water within bags of water. So eukaryotic cells and prokaryotic cells do have some things in common. We're going to be focusing on the eukaryotic cells, but just to kind of go over both of them at the moment. The basic structure and functional unit of both is a cell. The only organisms that are prokaryotic are the ones in the domains bacteria and archaea. Protists, fungus, animals, plants, all consist of eukaryotic cells. So most of the creatures that you would see in your local zoo or when you walk outside, they are of the type that have eukaryotic cells, meaning they have all these little organelles that we're going to look at. Even though we're going to focus on the eukaryotic cells, let's go ahead and compare the two just to make sure we understand between both prokaryotic and eukaryotic. Below is a list of features that are applicable to all cells. So whether you're a bacteria cell or a human cell or a dog cell or a oak tree cell, all cells have a plasma membrane of some kind. All cells have a liquid inside called cytosol. All cells have chromosomes, DNA, genetic material, and all cells have ribosomes, which are the little organelles that make proteins. So of any living creature that's made up of cells, which all living creatures are, these cells all contain these things in common. Now let's look at the most simple ones first, just to kind of get them out of the way before we launch into the eukaryotic. Prokaryotic cells are characterized by having no nucleus. They also have no membrane-bound organelles inside of them. They do have DNA, but it's simply in an open area inside the cell called the nucleoid. They do have cytoplasm. They do have this liquid material, cytosol. Cytoplasm would be the cytosol plus everything else in there. And it is bound by a plasma membrane. Here is a drawing and then a picture. This is a transmission electron microscope picture on the right-hand side. Basically a section of the bacteria that they've sliced thin and then put on a slide. And then over here on the left, of course, is a drawing that kind of represents what you see here. So they do have a plasma membrane. Bacteria also have a cell wall similar to plants. Inside, they have their DNA material is in this area called the nucleoid, but it's just free-floating. There's no mitochondria, there's no other organelles, there's no Golgi apparatus, lysosomes, or anything like that. They do have ribosomes, which all cells have ribosomes to create proteins that they need. But it's a fairly simple creature. You'll see this one has some little hairs sticking out that we'll talk about a little bit later. And then it also has like some tails here called flagella to help it move around as well. So a fairly simple creature. This would be a prokaryotic creature. These are not the kinds of cells that make up an oak tree or a dog or a human body. The more complex cells that we're going to be studying are called eukaryotic, and they have a few characteristics that make them unique. Number one, their DNA is bound in a particular organelle called a nucleus, and that's surrounded by its own nuclear envelope. They have lots of these membrane-bound organelles, so we'll take a look at things like lysosomes and Golgi apparatus and endoplasmic reticulum. They have cytoplasm, which is the liquid material in between the plasma membrane and the nucleus. And eukaryotic cells tend to be quite a bit larger and more complex than the prokaryotic cells. So why don't we start on the outside, kind of work our way in. The outside of the cell is made up of this plasma membrane, and it's basically kind of like the guard. We're going to compare a eukaryotic cell to, say, a city. And if the eukaryotic cell is a city, well, the plasma membrane would be like the outer wall of an ancient city. and perhaps the guards that stand guard at that outer wall, they decide what comes in, what is able to go out. So it's a selective barrier. In other words, it doesn't just allow everything in and everything out. It kind of decides what's going to come in and what's going to come out. That could be oxygen, nutrients, waste product is going to move back and forth between. This is made up of a double layer of phospholipids. If you'll think back when we were talking about those large macromolecules, the phospholipids, remember, were a type of lipid material where it had these fatty acid chain tails, two fatty acid chain tails, and then a phosphorus end. So it had a hydrophilic head, which was attracted to water, and then it had two hydrophobic fatty acid tails that were hydrophobic. In other words, they were not easily mixed with water. And so when they got together, they formed this bilayer. So here you see a picture. Here's actually a transmission electron microscope picture. And then, of course, down here is a drawing. And then so you see the hydrophilic heads make up the outside portions. And then the hydrophobic tails make up the inside portions. So in this picture here, you might imagine that this blue area is outside of the cell, and the pinkish tan area is inside the cell. And this is the cell membrane that makes up that outer wall. One great question that always seems to come up when we're talking about cells is, if you think about different sized animals, like you compare a mouse and an elephant, a great question that you often have from elementary school students is, does an elephant have larger cells than a mouse does? And the answer is no. Larger creatures don't have larger cells, they just have more of those cells. And so then the next question would be, well, why not? How come cells can't be extremely large? How come elephants don't have cells that are the size of beach balls and mice have cells, the size of a pinhead or something like that? And the reason is because there are certain metabolic requirements that constrain the size of a cell and actually the size of a living creature as well. And it has to do with surface area and volume ratio of the cell. To be alive, the cell has to move materials in and out. And the larger the cell becomes, the less surface area to inside volume there is. And so you get to a point where if a cell is too large, it doesn't have enough surface area and can't move materials in and out fast enough to take care of the huge volume of material inside of it. And so a good way to look at this is just like with a square cube. So here, what we see is as we increase the size of that cube, there's a ratio that increases as well. Here we have three examples, and we're going to pretend that these little cubes represent cells. So let's say that it was the way that you think it might be, that mice have little bitty cells, like this little square, and elephants have very large cells, like this larger square here. Well, right here you see a surface to volume ratio, which is very important. The cell, to do everything that it needs to do, has to have a very high surface to volume ratio. So if you have small cells like this, and only one of them, it does have the correct ratio. But the larger the cell becomes, the volume inside increases more than the surface area of the outside. So this ratio begins to fall. And eventually you would get to a point where the cell doesn't have enough surface area to take care of all the volume inside. So what's a way around that? Well, you could have the same size of this large block, but divided into many, many smaller blocks. And then, see what happens? You have the same total volume. These two have the same total volume, but because you've divided it up into many, many small blocks, you have a much larger surface area, and that ratio is back to where it needs to be. So you can have a large creature, but that large creature is not made up of large cells. It's made up of more of the small cells. And that gets back to this important ratio of surface area to volume of the cell. So let's take a look at these eukaryotic cells, the cells that make up your body, the cells that make up the trees you see outside and your dog at home. On page 100 and 101 of our textbook, there's a great kind of drawing or overview of the eukaryotic cells. And we already said that eukaryotic cells have these internal organelles that have their own little membranes. So basically it's a bag of water with smaller bags of water inside. And most plant and animal cells tend to have the same organelles. So if we were to take a cell from your body and compare that to the cell of an oak tree, we would find a lot of similarities. There's a couple of great movies. I believe you can view it right here in this PowerPoint if you just click on the video here, click it on it to play, or if you'll go to the notes area of Chapter 6, you'll find these videos. These are called Bioflix movies. There's one for a tour of an animal cell, and there's one for a tour of a plant cell. And these are really cool animations that kind of show you what's going on inside these very complicated and amazing eukaryotic cells. Here's a nice diagram or model of a basic animal cell. Could be any particular animal cell. It shows pretty much all the organelles we're going to take a look at. I know it kind of looks overwhelming when you see them all together, but we're going to break it down and look at each one of these individual parts, kind of talk about what is their purpose, why are they in the cell, what do they do, what is their structure, and then what does that structure allow them to do. So you see all these different organelles. Probably the most prominent one, of course, is the nucleus right here. And we'll break that down and talk about it as we move along. Here are some actual pictures, photographs, microscopy work. So here we have some animal cells taken from the lining of a human uterus. Here we have some yeast cells, and this is a colorized scanning electron microscope picture. And then we have a single yeast cell, a cross-section of that yeast cell. And this again is a colorized, but this one is from a tunneling or transmission electron microscope picture. Here's a great model of a plant cell. You'll find that it has very much the same parts as an animal cell. A couple of differences, of course. You're probably familiar with them as far as the cell wall and the chloroplast, but most of the parts are actually the same. So that, again, goes to kind of show you this evolutionary heritage that we share in common with all living creatures. Here are some great pictures, some microscope pictures. Here's a colorized transmission electron microscope of a plant cell from duckweed, which is a common pond floating material. Here is a particular little protist. Remember, the protists are kind of up in the air right now as far as dividing, how they're divided. They used to be just called protists, but there's a lot of different kinds of protists, and scientists are currently kind of subdividing that category. But here's a little protist, a very cool colorized SEM. And then here's a colorized TEM of that same protist showing the chloroplast, cell walls. Kind of makes him more like a plant. There are plant-like protists, and then there are animal-like protists. This one happens to be more plant-like. Now that we've kind of made our way inside the cell, we've gotten past that cell membrane, we're into the cell. Probably the first place you want to go is to find the leader, right? So when the aliens land, the first thing they ask is, where is your leader? Take me to your leader. So if you wanted to find the leader of the cell, or the leader of our little cell city, you would go to the president or the mayor. In this case, that would be the nucleus. So Concept 6.3 says, the eukaryotic cell's genetic instructions are housed in the nucleus and carried out by the ribosomes. So we're going to kind of compare our cell to a city, and if that's the case, well, the nucleus would be like City Hall, and the DNA would kind of be the boss. He would be the foreman or the person handing out all the instructions. The nucleus contains most of the DNA in a eukaryotic cell. We're going to find there's actually some DNA located elsewhere, but for the most part, the DNA is located in the nucleus. And then these ribosomes, which carry out the instructions of the DNA, they are throughout the cell in a couple different spots, and they make proteins. So we are going to find that there is some DNA, some genes located in mitochondria and chloroplast, but for the most part, the instructions are located in the nucleus. So heading to the nucleus contains most of the cell's genetic information, and it's usually the first organelle that you see when you look in the microscope. It tends to be fairly large. You could consider this like the brain or city hall of the cell. It's enclosed by what we call a nuclear envelope. That encloses the nucleus and keeps it separate from the rest of the cell, so it kind of has its own little structure or palace. The nuclear membrane is actually a double membrane, so this envelope or membrane that covers the nucleus is actually two layers, a double layer, kind of like the cell membrane is, and there's a tiny little bit of space between those layers. So it's a lipid bilayer, much like the cell membrane that encompasses the whole cell is. Here we have some great microscope pictures of a nucleus. This would be from a scanning electron microscope showing kind of the surface of the nucleus. The nucleus does have small openings called pores, and you can see those pores right here. Those pores allow certain materials in and out. We'll talk about that in just a little bit. Inside the nucleus, you'll find the genetic material. You have long strands of DNA wrapped around proteins. This is called chromatin. Right here is chromatin. And usually it's kind of diffuse. In other words, if you were to look at the nucleus, you would see the chromatin, but it would look like threads, just all kind of matted together. When the cell gets ready to divide, those threads actually bunch together and you see the chromosomes. So the chromosomes are basically just the gathered chromatin all together there. So that's all the genetic material, and then you can see it's surrounded by other material we're going to look at, including the endoplasmic reticulum. There are pores that lead in and out of the nucleus, similar to, say, like the pores in your skin, tiny openings that regulate the entry of certain materials, and we're going to talk about those. And then there's something called the nuclear lamina, and the lamina is kind of like the structural framework that kind of holds the nucleus and gives it its shape. In the nucleus, the DNA, which is the instruction material, is kind of organized into these units that we call chromosomes. And each chromosome is a long single-stranded DNA wrapped up together with proteins that kind of hold it together. The DNA and the other proteins of the chromosome all together are called the chromatin. So if we kind of look at this picture here, DNA in long strands is actually wrapped around proteins. We call that chromatin. And then it's in units or portions that we call chromosomes. So this chromatin or chromatin tends to be kind of in a diffuse state when the cell is going about its normal business. But as the cell prepares to divide, the chromatin or chromatin condenses and it forms these discrete chromosomes that are usually quite a bit more visible and able to be seen through a microscope. As the cell prepares to divide, it will double up these chromosomes, so it's doubling up its genetic material and then divides. Of course, we'll talk about that a little bit more later. There's another part of the nucleus that we're going to talk about called the nucleolus, and that is coming up. And that's an area of the nucleus where ribosomal RNA, which is called rRNA, is synthesized. And we'll talk about what ribosomal RNA is. It's another part of genetic information that helps create an important part of the cell called the ribosome. Just for FYI, our own information, different species of plants and animals have different numbers of chromosomes. Like you're probably familiar with the fact that humans have 23 pairs. But the number of pairs of chromosomes has really nothing to do with the complexity of the creature, or there doesn't seem to be any particular pattern. For example, humans have 23 pair of chromosomes. You get 46 total chromosomes. You get 23 from mom, 23 from dad, and they pair up. Dogs have 39 pair, cats have 19 pair, but there are other creatures, like there are some simple plants that have far more than humans, and then there are some things like dogs that have more and cats that have less. So there doesn't seem to be any direct correlation between the number of chromosomes and the complexity of the creature itself. So we said there in the nucleus we have the DNA, the instruction material, kind of like the boss of the whole place. And then we also have the production of workers called ribosomes. Ribosomes are some of the smallest organelles within the cell, but they're perhaps some of the most important. They are made from ribosomal RNA and protein. So remember that portion of the nucleus that I just mentioned called the nucleolus? That's where ribosomal RNA is created. And this organelle called a ribosome is made up of this ribosomal RNA and protein. And these guys are the workers, or you could consider them, I guess, the miniature factories of the cells, the construction crew. They're the ones that carry out the instructions given by the DNA, and they're the ones that create the proteins. And the proteins, remember, are the building blocks for everything else, the building blocks for you and your cells and your body. So ribosomes carry out protein synthesis in two different places. Some of these ribosomes are just floating free out there in the cytosol. So just out in the liquid part of the cell, they're floating around freely, doing their job. And others of these ribosomes are attached to a particular part of the cell, either to the endoplasmic reticulum or the nuclear envelope, the nuclear membrane itself. So you can have free ribosomes floating around in the cytosol, or you can have what we call bound ribosomes that are either attached to a part called the endoplasmic reticulum or they're attached to the actual nuclear membrane, nuclear envelope itself. Here we have a cool TEM of the endoplasmic reticulum and showing the ribosomes. The ribosomes are just the little dark dots that you see right in here. So here are some free ribosomes floating around in the cytosol, and then you have this line as part of the endoplasmic reticulum, and you have some of those ribosomes that are stuck to or bound to that. The ribosome itself is made up of like a two-part structure. It has a large unit, subunit, and a smaller subunit. And we'll talk about that a little bit more as we go. So the ribosomes are kind of like the workers of the cell. They are the ones churning out the proteins based on the DNA instructions, and it's those proteins that are then used to build everything else. So we're going to take another pause right here. So far we have been to the city walls, the city gate. We have entered through the cell membrane, which is a phospholipid bilayer. We've headed to the city hall to meet the nucleus, or I guess you could say the main controlling portion of the city. Inside the nucleus, we know that's where the DNA and genetic material resides primarily. And then we also have this area where ribosomes are created. And ribosomes, although small, tend to be some of the most important parts of the cells because they're the ones making the proteins. We'll go into more detail in just a second. Take a break. Go get some iced tea or something to drink. Come back and join me. Thanks, guys. Bye-bye.